the epipium. In this form, they are resistant to desiccation, freezing, and digestive enzymes, and such as play an important role in colonizing new habitats or in the re-establishment of an extinguished population after favorable seasonal conditions. This is the picture of Daphnia, where you can see the midgut, eggs, epipium, apical spine. These are the hindgut, anus. These are the carapace, foregut, rostrum, antennulae, labrum, mandibles, and maxillae. Now, what is the importance of Daphnia? What are the nutritional values of Daphnia? The nutritional value of Daphnia depends strongly on the chemical composition of their food source. However, since Daphnia is a freshwater species, it is not a suitable prey organism for marine organisms because of its low content of essential fatty acids and in particular N3-HUFA. Furthermore, Daphnia contains a broad spectrum of digestive enzymes such as proteinases, peptidases, amylases, lipases, and even cellulase that can serve as exoenzymes in the gut of the fish larvae. Daphnia are filter feeders. The filter single-celled algae and other foods. They can be cultured in water from the taps and culture green waters. Green water can be produced in large aquarium or small tanks, garbage tanks in the backyard. Suitable water can be filled in small containers and used for culturing Daphnia. Care should be taken to see the temperatures do not fluctuate rapidly as they are highly sensitive to green water. If they're not overfed and if the culture water is maintained, changing partially as and when required, can keep the stock of Daphnia healthy and growing tubes of 25-gallon capacity are more suitable to raise Daphnia. They can be fed with other food items like sweet potatoes, yeast, and water solutions. Now let us study about moina. Moina are a type of freshwater zooplankton that can also be used as a live food source because they are nutritious and ideal for feeding small fish, fry, and crustacean larvae. Moina are also one of the easiest live foods to culture at home in small containers. With a basic setup and proper care, aquarists can produce a regular supply of moina to feed out to their fish. Now, how to do the stock culture? Pure stock of moina is developed for inoculation in the large-scale mass culture tanks for this. So at first, moina is obtained by collecting from the natural labs or tanks. Then few individuals are picked up by dropper and distributed and disturbed one in each set tube of 20 milli capacity containing 50 milliliters of 250 ppm. A single moiner produces 8 to 10 offsprings in about 24 hours. These offsprings from the test tubes are then transferred into 2 liters capacity cylindrical glass jars. The tank stock of moina so obtained is inoculated in the large tanks for mass culture. So mass culture, depending on the requirement of moina, the culture tanks are selected. Mass culture of moina is undertaken in 15 to 20 meter cube rectangular tanks so as to meet the hatchery requirement regularly. The selected tanks are thoroughly cleaned and filled with filtered fresh water and constantly aerated. The tanks are covered with closed mesh nets to avoid the interference of dragonfly. If the dragonfly are in contact with the tank water, they lay the eggs and the eggs hatch and become nymphs, which voraciously feed on the cultured cultural moina. In this, there are methods. The first method is the tanks are treated with grounded oil cake, single superphosphate, and urea 75 ppm, 25 ppm, and 8 ppm, respectively, after fertilization. Phytoplankton is inoculated for stock culture if available. If not, the culture tank is directly inaugurated with pure stock culture of moina at 40 to 50 individuals per liter, depending on the availability of the stock culture. If the stock culture is less, the initial inoculation rate is reduced as per the convenience. Moina grows rapidly, feeding on phytoplankton. Phytoplankton blooms, which are developing by utilizing fertilizers applied and the sunlight and the organic matter and attains a peak density of 20,000 to 25,000 individuals per liter in 5 to 7 days. The density and the culture. So the number of individuals inoculated initially after attaining peak density, regular harvesting is done to feed the prawn larvae. Now, how to feed the moina? In the wild, moina eat microalgae, bacteria, and detritus. As a matter of that, moina are one of the few zooplankton which can eat the blue-green algae. Moina eat between 50 to 200% of their body weight in food particles daily. So, feed the culture small amounts of powdered food every day. Add a pinch of fertilizer or dried manure to stimulate algae growth, which moina will graze on. Green water is also great for culturing moina. In this pic, we can see how a moina looks with the antennae. Moina reproduces extremely fast. In 1 to 2 weeks after setting up, the culture can have 100-fold increase in biomass, turning the water greenish brown. Thus, it is time to harvest moina. It is better to do this during daylight hours when moina are suspended in the water column. Turn off the aeration and allow the food particles to settle. Take a fine mesh net or a strainer. Nets with mesh around 0.3 mm work best to retain moina. The coffee filter also at times we can use. Scoop the net through the 20 to 30% culture while letting water pass through. Rinse harvested moina in a bowl of clean, dechlorinated water. Jars are small bags to store harvested moina for feeding out over the next 1 to 2 days. Next, coming to cyclops. So, it's a genus of small freshwater crustaceans. We call them as copepods, characterized by a single eye spot on the head segment. Cyclops also feature antenna, a segmented body, 5 pairs of legs, and a divided tail called a furca. Cyclops has a cosmopolitan distribution in freshwater, but is less frequent in brackish water. It lives along the plant covered banks of stagnant and slow flowing bodies of water, where it feeds on small fragments of plant material, animals such as nematodes or carrions. It swims with characteristic jerky movements. Cyclops has the capacity to survive unsuitable conditions by forming a cloak of slime. Average lifespan is about 3 months. This is the Nauplius, and the adult Nauplius looks like this. That is, we can call it as Copepoda or Cyclops. Copepoda. These copepods have 6 Nauplius stages, followed by a stage called the copepodid, which has the same number of body segments and appendages in all copepods. The copepodid larva has 2 pairs of unsegmented swimming appendages and an unsegmented hind body comprising the thorax and the abdomen. There are typically five copepodid stages, but parasitic copepods may stop after a single copepodid stage. Once the gonads develop, there are no further molds. The nauplius stage. It is characterized by consisting of only three head segments, which are covered by a single carapace. The posterior body, when present, is unsegmented. Each head segment has a pair of appendages, the antennules, antennae, and mandibles. This larval stage has various lifestyles. Some are benthic, while others are swimmers. Some are feeding, while others are non-feeders. Harvesting cyclops. Harvesting cyclops is a relatively simple process. You need a fine mesh net or a sieve. The net can be gently passed through the culture, collecting the cyclops as they swim into the net. The collected cyclops can then be rinsed with fresh water and transferred to a separate container for further use. If you look at the cyclops anatomy, antennule, antennae, ventral nerve cord is there, this is the thoracic region. You find this abdomen. These are the egg pouches, genital segment, gut, ovaries or testis, eye, and head region. Now, what is the basic setup for cyclops culture? Setting up a culture for cyclops is relatively straightforward and requires only a few basic materials such as small culture container, plastic or a glass jar, or an aquarium, aeration system, air pump, bubbler stone, etc. Light source, example, fluorescent light, natural light. Culture food, that is spirulina, phytoplankton, rotifers, other small copepods, etc. Now, how to proceed with that? Fill the culture container with a clean and dechlorinated water. Add an aerated system to the culture container, such as an air pump. Place the container in a well-lit area, either under a fluorescent light or in a spot that receives natural sunlight. Although cyclops do not require light too much, their prey does. Thus, providing adequate light is also important. Let algae and biofilm grow in the culture container before adding cyclops. You need to wait for at least a few days. Do not drop them off right away. Add a starter culture of cyclops. Then feed the cyclops with a culture medium such as yeast, wheat flour, or oatmeal. These foods provide the cyclops with the nutrients they need to grow and reproduce. Start with a small amount of food and gradually increase the amount as the cyclops population increase. Overall, setting up a culture of cyclops is relatively simple and can provide a reliable source of live food for your fish. By following these basic steps and monitoring the culture regularly, we can ensure that our cyclops culture remains healthy and productive. Now, the size should be the optimal tank size for cyclop culture can vary depending on the number of cyclops you want to culture and the space you have available. Generally, a container that can hold at least a gallon, that is 4 liters of water, is sufficient for culturing a small population of cyclops. Now, temperature should be generally most species of cyclops thrive in temperatures between 73 to 78 degrees Fahrenheit, that is 23 to 25 degrees centigrade. Sudden fluctuations in the temperatures can stress the cyclops and lead to decreased growth and reproduction. Lighting. Even though they are sensitive to light, cyclops are not photosynthetic organisms and do not require specific lighting for growth and reproduction. So maintaining adequate oxygen levels and providing proper aeration is important for the health and reproduction of the cyclop cultures. Cyclops require a constant supply of dissolved oxygen to survive, and the levels of oxygen in the culture can fluctuate rapidly due to changes in temperature, feeding, and other factors. The optimal oxygen level for cyclop culture is between 5 to 8 mg per liter. Feeding cyclops culture. So, contrary to popular belief, cyclops are non filter feeders. In reality, cyclops are mostly predatory creatures that primarily feed on simple organisms such as phytoplankton, rotifers, other copepods, young mosquito larvae, eggs, and even small crustaceans. In addition, cyclops are known to be efficient predators able to consume slightly larger prey such as daphnia or even tiny fry. So this is cyclopoids predominantly these are predators and these are herbivores. Calnoids are herbivores, cyclopoids are predators. Now, these are the different stages. Cysts. These are dormant eggs that can survive harsh conditions. Nauplii, after hatching from the cyst, the larvae are called nauplii. Juveniles, as they grow, they molt and develop into juveniles. Adults, adults can live for several months and reproduce by producing new batches of eggs. So these are the dormant cysts. After hatching, these are the naupliar stages. Then male and female, these are the juveniles, and they grow into adult male and adult female. Next, coming to Artemia. Artemia is also known as brine shrimp. It's an aquatic marine animal found in high salinity seawater. Brine shrimps are the preferred live feed in the fish and shrimp hatcheries. Brine shrimps are highly nutritious, which helps fish and shrimp larvae to grow faster. Fish and shrimp larvae are very small in size and cannot feed them the commercial feeds that are available on the market. So, natural feeds are preferred, that is, phytoplanktons and zooplankton. Brine shrimps are being used as live feed for a long time in hatcheries. Artemia eggs are brine shrimp eggs, are widely available online as well as in offline aquaculture stores. Brine shrimps are one of the best live feed sources for shrimp larvae, fish larvae, and aquarium fishes. That is the reason the Artemia eggs are very high in demand as one of the best live feeds. Cultivating Artemia is also known as brine shrimp, is rewarding for both aquaculturists and hobbyists. It's great for starting a Artemia culture in your aquarium or making nutritious Artemia nauplii for feeding. So this guide will help us through very step by step from the beginning to the end. First, get high quality Artemia cysts from a trusted source. Then set up your hatching system. Pick a good container, mix the water right, add air and light it up. Soak the Artemia cysts in the water for 1 to 2 hours to hydrate them. Start the hatching by adding light and air to the cysts. Watch the hatching closely and adjust the water as needed for the best conditions. Collect the Artemia nauplii when most have hatched. usually in 24 to 36 hours. Move the Artemia napoli to a new container for growth. Keeping your Artemia culture healthy means watching the water, temperature, and salt levels. Regular checks and adjustments will help your Artemia grow strong. This way, you will have a steady supply of live food for your fish. Changing up the feeding routine can also help your Artemia culture. Try giving them a mix of foods like microalgae such as Nanochloropsis or Tetraselmis, yeast or baker's yeast. Commercial Artemia feed supplements. Offering a variety of foods ensures your Artemia napoli get a balanced diet. This leads to healthier and more nutritious live Artemia feed for your animals. Using the right feeding and enrichment methods is key to a good Artemia culture. By following these tips, you can give your aquatic animals a top notch live Artemia feed. So, this supports their health and growth. After that comes decapsulation. For this, add lime, that is calcium oxide CaO, at the rate of 12.5% of the weight of eggs. Stir until mixed thoroughly. Then add bleaching powder at the rate of 27.5% of the weight of Artemia eggs and keep on stirring until the substances are mixed thoroughly. This stimulates the reaction of the bleaching powder in removing the shell of the eggs. The procedure must take place at a controlled temperature of 40 degrees centigrade. Do not let the temperature exceed 40 degrees centigrade. It can be maintained by adding more ice. Stir for 5 to 8 minutes until the temperature stays steady. This shows that the reaction of the first step of decapsulation has been completed. Then add some more ice to decrease the temperature to 30 degrees centigrade and then continue with a second decapsulation stage. In the same manner as the first stage, adding lime and bleaching powder in the same amounts. Stir for 5 to 8 minutes until the decapsulation reaction is complete. The color of the eggs will change from dark brown to white. Leave them at that stage until the white color gradually becomes orange. The whole procedure, including both stages, will now have taken 10 to 16 minutes. These are the Artemia cyst. We can get it in the shops offline or online. Next, coming to diatoms. Diatoms are small organisms found in oceans, waterways, and soil all around the world. They belong to a group of algae called microalgae. These tiny creatures play a big role in our environment. They produce a lot of oxygen, about 2 to 50% of Earth's total, which is really important for us to breathe. They also take in a lot of silicon from the water they live in, and they make up almost half of the organic material in the oceans. If you look into the diatoms, they are single celled organisms and they can live alone or in groups. They come in various shapes and sizes, ranging from 2 to 200 micrometers. They produce by splitting into new cells, and they can double in number about every day if they have enough nutrients and sunlight. One interesting thing about diatoms is that they have a harder outer shell made of silica, which is a type of material found in glass. These shells can be really colorful and are sometimes called jewels of the sea. Diatoms can move around passively in water currents and tube and turbulence, and sometimes even have flagella to help them swim. Diatoms are like tiny factories that use sunlight to make energy through a process called photosynthesis, just like plants. They are a type of plankton, which are tiny organisms that float in the water. Diatoms are an important part of the ocean's food chain. providing energy for many other creatures, from small fish to big whales. Scientists study diatoms to understand more about the environment, and they can tell a lot about water quality from studying diatoms. These little organisms are also used in various ways, like in water filters and even as dynamite stabilizers. Despite their small size, diatoms have a big impact on our world and are truly fascinating to explore under a microscope. You can see the picture of different shapes of the diatoms, how beautiful and colorful they are looking. Now, how to isolate them? These are the techniques: manual isolation, automated isolation technique, and if you look into manual isolation technique, algal cells were isolated using micropipette. However, this method required refinement as it gave bacteria infected diatom cultures, although of reduced population, a detrimental factor for any pure culture. Use of pasture pipettes in the isolation of specific diatoms. This technique was subsequently refined to avoid bacterial contamination by picking up single cells of filaments with a capillary pipette. An exhaustive description of the pasture pipette technique is given in algal culturing techniques. Agar plate method combined with antibiotic treatment. Agar plating method is used for the isolation of diatoms infested with bacteria, algae, etc., thereby acquiring axinic culture of diatoms. Generally, higher concentrations of antibiotics combined with short term incubations were more efficient than using low concentrations for longer periods. Algal contamination. So, how algae also get contaminated? Diatoms are first concentrated by continuous centrifugation on sonication or to avoid clumping and then isolated by a micropipette onto an agar plate containing the required media. This is then followed by repeated subculturing and streaking the colonies into agar plates. Next, let us study about cyanobacteria. Cyanobacteria is also known as blue-green algae. They are microscopic organisms that exist naturally in all types of water. These single-celled organisms can exist in fresh, brackish, and marine waters. These organisms use sunlight to produce their food. Cyanobacteria thrive in warm, nutrient rich settings, forming blooms on the water surface. Cyanobacteria are larger than bacteria and possess chlorophyll A. Some kinds contain specialized terminal structures known as heterocysts. Heterocyst bearing cyanobacteria are all aerobic photodiazotrophic. Blue-green algae are sensitive to light, salinity, temperature, and nutritional change. Now, what are the functions of cyanobacteria? They have several functions that can help us in many ways, like crop production. Cyanobacteria improve crop production by acting as biofertilizers, improving soil conditions, mineralizing organic molecules, stimulating plant growth, and acting as a biological pest control agent. Then nitrogen fixation. Terrestrial cyanobacteria species improve soil fertility by fixing atmospheric nitrogen, binding soil particles, retaining moisture, and avoiding erosion. Blue-green algae or cyanobacteria fix atmospheric nitrogen through biological nitrogen fixation. As photosynthetic organisms, they do not compete with agricultural plants or soil microflora for carbon and energy. Soil fertility. So, nitrogen fixing plants like Anabina, which grows in rice fields, with the floating water fern azolla, improve soil fertility and enhance rice output. What are algae? Algae exist in environments ranging from oceans, rivers, and lakes to ponds, brackish waters, and even snow. Algae are usually green, but they cannot be found in a variety of different colors. For instance, algae living in snow containing carotenoid pigments in addition to chlorophyll, hence giving the surrounding snow a distinctive red hue. What are the characteristic features of algae? Specific general characteristics of algae are common to plants as well as animals. Algae are photosynthetic organisms. Algae can be either unicellular or multicellular organisms. Algae lack a well-defined body, so structures like roots, stems, or leaves are absent. Algae are found where there is adequate moisture. Reproduction in algae occurs in both asexual and sexual forms. Asexual reproduction occurs by spore formation. Algae are free living, although some can form a symbiotic relationship with other organisms. Now let us see various types of algae. Examples. First one, charophyta. Now what are these charophytes? Let us study a little bit about this charophyta. Charophyta, chlorophyta, chrysophyta, cryptophyta, cyanophyta, dinophyta, euglinophyta, haptophyta, and xanthophyta. If you see charophyta, they are called as stonewarts, occur in ponds attached to the bottom by rhizoids. Examples, chara and nitella. Next, second one, if you see chlorophyta. What are chlorophytes? So these are a large and varied group of green pigmented algae which store energy in pyrenoids and as starch. These forms include unicellular, filamentous, and colonial varieties. Examples are chlorella, spirogyra, cinedesmus, edogonium, volvax, etc. Next, the third one is chrysophyta. These are also called as golden brown algae. Example, Cinera euroglina. Next, cryptophyta. These are unicellular with two slightly unequal flagellae at the apical end. Example, Chelomonas and Cryptomonas. Next one is Cyanophyta. These are also called as blue-green algae, also called as cyanobacteria. They have no nuclei, and now these are classified among us the bacteria because their lack of a nucleus distinguishes them from all other algae. Now, examples for cyanophyta or cyanobacteria are spirulina, anabina, oscillatoria, and nostoc. Next is dinophyta. Dinoflagellates are unicellular biflagellate with flagellae in two different planes. External cell wall often features sculptured plates. Frequently, the main organism in toxic algal blooms. Example, Dinophysis acuta. Next one is Euglinophyta. These are unicellular with single obvious flagellum. They usually have a red eye spot green plastids, and paramylon granules called pyrenoids. Example Euglena, Pacus, and Trachelomonas. Next is Haptophyta. These are also called Haptomonada, a smaller group of unicells bearing calcareous plates, that is scales and coccoliths, and a haptonyma visible only with an electron microscope in most cases. So these are primarily marine and very few are freshwater genera. Next one is the last one is Xanthophyta. These are yellow green algae. The color is due to chlorophyll and carotenoid pigments. Maybe unicellular, filamentous, colonial, or siphonochus in form. Example, Tribonema botrydum. Next, coming to these are the six types of plant-like protists. See, brown algae, they have many plant-like structures, is the brown algae. And the second one is red algae. Most are multicellular seaweeds. Then the third one is diatoms. These are unicellular glass-like cell walls. Now fourth one, green algae. Fifth one, dinoflagellates. These are unicellular and they have armored algae. They are armored algae. Next, sixth one is euglenoids. Green unicellular algae and they have a eye spot. These are few more examples of red algae, brown algae and green algae. Red algae examples are Rhodymania, Pilophora, Deliceria. Brown algae example Macrocystis, Laminaria and Fucus. Green algae examples Nitella, Caulerpa and Codium. Let us look into desmides. What are desmides? So this desmid is a unicellular organism. It comes under the green algae family Mesotaniaceae and Desmidiaceae. Sometimes desmid placed under the family of order Gignematales. Desmides are usually developing in standing freshwaters. As it is a unicellular organism, it is impossible to view a single desmid, but can view the largest representatives in the naked eye. This green algae comprises 40 genera with 5,000 species. Desmids in food web. So desmid is a well-known aquatic species. It plays an important role in aquatic ecosystems. The desmids are the type of unicellular biomass algae. It acts as an important food source for many invertebrates. The desmids are the potential food source for zooplanktons like Cladocera and copepods. The desmids use planktonic mixed desmids as a source of food. The deep planktonic desmids are a rich potential food source. As the desmids has a low population density, it plays a minor role in the pelagic food web. In particular, where desmids grow in a submerged substrate as a green film that can be viewed through naked eye. They are acting as a feed for the large numbers by grazers like rotifers, worms, midge larvae, etc. Now let us study about spirulina. It is a dried biomass of cyanobacteria, blue-green algae, that can be consumed by humans and animals. The three species are Arthrospira platensis, Arthrospira fusiformis, and Arthrospira maxima. Cultivated worldwide, this Arthrospira is used as a dietary supplement or whole food. It is also used as a feed supplement in the aquaculture, aquarium, and poultry industries. Now, these are the 10 health benefits of spirulina. It protects from medicine byproducts. So, whenever we take any medicines, they release certain byproducts which might become toxic in the body and they might cause side effects. And they are full of vitamins and minerals. They are great for cell regeneration and they are very good for good protein source. They are easily digestible and they are considered as superfood, and they are antifungal, and they also are very familiar for anti-aging as an anti-aging product. It can fight cancer and it is an antibacterial also. So what we have understood is spirulina can be cultivated at a very small level and at very large commercial level also. It can be cultivated by mud pond method and in very large racecourse pond forms also. Hence, it is easily adaptable for poor village farmers as well as industrial farmers. So, many women self help groups were producing spirulina and marketing the powder, and also different value added products can be prepared from it. Spirulina can be used as protein supplement in poultry and livestock feeds also. Fish meal, groundnut meal, and soybean meal can be partially replaced by spirulina in the preparation of diets of fish, poultry, cattle, and domestic animals. Fish meal and groundnut cake in a commercial diet containing both protein sources may be replaced on an isonitrogenous basis with dried spirulina, that is 140 and 170 grams per kg. That is for the starters, and 120 to 128 grams per kg, that is for the finishers. We can use this for broiler chicks. Now, how to cultivate this spirulina? Spirulina grows in alkaline rich water. These tiny spiral coils thrive in highly alkaline medium and harvest the energy of sun to supply a treasure of bioavailable nutrients beneficial to human body. The blue-green color comes from the two pigments, that is phycocyanin and chlorophyll. For conventional agriculture, the land is required. The spirulina does not need fertile land for cultivation. Hence, it can be cultivated in rocky land or even terrace top of buildings also, which can be used for conventional farming. Then, from less area, rocky land or even terrace. The blue-green, the spirulina requires less energy input per kilo than soy, corn, or bovine protein. It uses less water per kilo of protein than any other food as the water is recycled back to the ponds after harvesting. Spirulina is a big oxygen producer that is even more efficient than trees and forests to absorb carbon dioxide and release oxygen. Hence, the cultivation of spirulina is very much eco-friendly. Now, with this, we have come to the end of the chapter. Just like let us look into the summary of this chapter. Phytoplankton and zooplankton can be found in large quantities in small area, and as they are consumed in large numbers by herbivores and carnivorous fish, many of them reproduce asexually to keep population numbers high. These plankton forms an essential part of an ecosystem and support as natural food in the culture system of carps. Phytoplankton harness new energy from sunlight, multiply in abundance, and constitute as the means of food to herbivores. whereas zooplankton are also formed as an important source of food for many species. So, in this unit, by taking a few examples of phytoplankton and zooplankton are drawn, and their utility and culture methods are highlighted to facilitate better understanding of these very important natural aquatic fish food organisms. Now, from this chapter, the frequently asked questions can be: write a short note on plankton, or write a short note on Daphnia, or write in detail about Daphnia, its sources, and how to cultivate Daphnia. And what is parthenogenesis? How parthenogenesis occurs in Daphnia. What is the nutritional value of Daphnia? And write a short note on Moina, or write in detail about Moina, how to stock the culture, how to culture Moina, how is it useful for fish. Now, what is the feeding for Moina? Write a short note on Cyclops. Write in detail about Cyclops, their larval stages. Explain in detail about the larval stages of Cyclops also, or how to harvest Cyclops. What are the stages of, you can mention all these: dormant cyst, hatching, nuclear stages, juvenile stage, female, male, how they are in the life cycle, you can draw and also explain. Next, coming to Artemia, explain in detail about the life cycle of Artemia, how to cultivate Artemia, and write in detail about the diatoms. Write the uses or advantages and disadvantages of diatoms. What is manual isolation technique? What is automated isolation technique of diatoms? What is agar plate method? Write in detail about cyanobacteria. Why are they called as cyanobacteria? What are the functions of cyanobacteria? And next, what are algae? Write in detail about the characteristic features of algae. What are the types of algae? Describe any three or any four types of algae. Then what are red algae, brown algae, green algae? Describe with examples. And finally, what is desmid? Explain in details about desmids. Explain in detail about spirulina, their advantages and disadvantages, or how they are useful as a good protein, and what are the health benefits of spirulina. So these are the questions asked frequently. Hope you all have understood. Thank you.